So now we will have Tatyana Makushok, and she will tell us about what's controlled microtubule organization and intracellular transport in Stentor, and she comes from the Basal lab.
Thank you for the introduction, and many thanks for the organizers for giving me this joy of being able to talk to you about Stentor that we love so much in the lab of Wallace Marshall at UCSF. And specifically, I would like to talk to you about my favorite two questions in Stentor. And those are basically how are microtubules organized and how they're dynamically controlled. And kind of related to that is how is intracellular transport happening in Stentor and what molecular motors are responsible for that. And the reason why I'm so curious about intracellular transport specifically is because the cell is extremely huge. It literally can extend to up to 1 millimeter, which is kind of crazy because actually just by looking at the culture, by eye, you can see how the cells change their shape. And as you can see on the images, they do change their shape quite a bit. So they can be in their attached extended form when they can create nice flows that brings food into the oral pouch. That you can see here. Or whenever something annoying happens to the cell, it just contracts into a tiny, tiny ball within a fraction of a millisecond. So it's a very efficient mechanism, and then it takes about a minute to extend back into this attached form. And the third type of kind of state in which you can see a Stentor is a free-swimming cell. And you can see it also looks a little bit different. And as you can see on this drawing, the cell has a holdfast, and on the opposite side, it has all sorts of oral apparatus structures. It has frontal field, membranellar bands that creates very strong flows that brings food into the oral pouch. Also, you can see that the nucleus, the micronucleus of Stentor is actually nodulated. And what you can see here as well is those stripes that are actually of the beautiful blue color. And that's why this species is called Stentor ceruleus. And from now on, whenever I'm going to say Stentor, I'm actually going to be talking about this species, which is the species we study in the lab. And so you can see the stripes on this drawing, but actually in between those blue stripes, there are clear stripes. And under the clear stripes, you have this beautiful arrangement of microtubules that run from the kind of mouth region of the cell to holdfast. And the reason why the cell is so spherical is because it contracts when you fix it. So this is a tail region here. And you can see that in addition to the beautiful arrangement of microtubules that create all of this very complex structure, just the body of the center is actually covered with those pretty much parallel rows of microtubules. And when you look closely at them, they are not just bundles, they're actually very interesting structures. They are basically stacks of ribbons of microtubules. So they're very relatively flat sheet of microtubule formed, and then they're being stacked into the stack of ribbons. And this structure allows for the attachment of the cilium on that, obviously is very important for Stentor since it's a ciliate. And so I'm very, very fascinated by the structure, and I would love to basically to answer those questions during my time as a postdoc in the lab is, at least like try to answer part of these questions, is what proteins actually holding those microtubules together in those structures, and which proteins allow the cell to dynamically control the structure, because as you have seen, it changes shape very dramatically, and it can bend, etc. And all of this requires obviously like sliding of those microtubule ribbons past each other, which is a very interesting process. And secondly, obviously once you think about microtubules and the cytoskeleton, you immediately think about molecular motors, so I would love to know what they do in Stentor as well. And in terms of the methods that people use in Stentor, Stentor has been studied for a very long time, for more than a century, and specifically a set of people were very much interested in using Stentor as a model for kind of unicellular development. And a lot of microsurgery has been done in Stentor. So this is one of my favorite examples, when the cell is basically cut in two, and then its tail is being bent on the rest of the cell. And as you can see on this drawing, eventually after a number of hours, the cell will perfectly reorganize into its normal shape. So Stentor has this amazing ability to reorganize its cytoskeleton and also to regenerate after being very drastically perturbed, as in this example, or being cut in pieces as well. And Mark Slobodnik, who used to be a grad student in our lab until recently, he established he brought modern biology into the Stentor field by establishing RNA interference protocol for Stentor by feeding them bacteria. And on this example, you can see that when Mark performed RNA against beta-tubulin, instead of those beautiful parallel arrays of microtubules, first of all, you can see that the cell shape is very weird. And also you can see on the immunofluorescence that microtubule rows are basically broken and disorganized in many different ways. And this question of how is polarity established in Stentor in general, and how is the polarity and proper organization of the cortical, proper organization of the cortex is established, is definitely very interesting for us. And the only information that is published so far, that's again work by Mark Slobodnik, where he has performed RNA against MOB1, which is a conserved polarity protein that you can find in many different species. And as you can see, again, instead of parallel microtubule rows, you still have parallel microtubule rows, but they're disorganized in very drastic ways. You have all sorts of defects in the organization of microtubules. And also the cells grow bigger. That's why it's actually two images combined here. So I would love to take this work further and to try to find more proteins that are required for organization of cortical rows in Stentor. Since we have RNA working, I'm very excited about using it. And specifically, I'm performing the RNA screen in Stentor right now. And thanks to amazing colleagues in the lab, they actually sequenced the genome of Stentor and assembled it. So I was the lucky person to be able to use the genome. And so I picked 300 genes, everything I could think of in the context of cytoskeleton and molecular motors. And just went ahead and already screened quite a bit of them. And what I would like to talk to you about today is my two most prominent phenotypes so far, and my favorite ones. So one of them is actually protein phosphatase 2A. So there are actually 4 different types of protein phosphatases. There is protein phosphatase 1, and there's protein phosphatase 2A, 2B, and 2C. So all of them are extremely important, and they're involved in pretty much every process that you can think of in the cell, in other cell types. It's kind of amazing. They interact with like lots of different molecules, and they control pretty much every process, as I just mentioned. In terms of the structure, PP2A is composed of 3 different subunits. There is a scaffolding subunit, there is a catalytic subunit, and there is regulatory subunit that is actually called B subunit. And the RNA against this specific regulatory subunit B gave me this. So instead of the normal Stentor shapes that you have seen before, I have observed those cells. And so there's multiple problems with those cells. First of all, they have this very folded surface, which is not at all present in normal Stentors. So somehow either because there are some problems with cortical organization or some membrane related processes are going on, cells become folded basically, and more folded over time. Also, there is a trouble with the mouse region, because mouse just basically gets kind of disappears over time, gets resorbed. Like in this cell as well, it just basically disappears. And over time, those phenotypes, the phenotype becomes even more drastic. You entirely lose the mouth, you entirely lose normal cell polarity, and the last stages, the cells just look wrinkled and they continuously contract. And unfortunately, this is a lethal situation for the cell. When I looked at what happens to microtubules in this case, to my big satisfaction, I observed a range of problems with microtubules. So as you can see, this is an early stage of the RNA experiment, and this is a later stage. And you can see that the mouth region, the frontal field, is much, much, much smaller in this case. And the microtubules are generally broken, so the arrangement of the rows is not correct in different regions. So I would love to understand how is this even possible, and how exactly why would the mouse be resorbed in such a dramatic way over time. So in addition to the RNAi, I inhibited PP2A with ocadaic acid, and actually using quite high concentration of it. And this drug is known to inhibit PP2A and PP2B. Sorry, PP1 and PP2A. And what you can see as well is that over time you see the folds on the surface of the cell. So it does kind of correspond to the phenotype I observed with RNAi. And also over time, you see those interesting kind of kinks on the surface of the cell, which I assume means that those will be the regions where cortical rows basically don't connect well, and there's some sort of cortical row problem in there. I will have to still check which of those phosphatases is actually inhibited with ocadaic acid, because in paramecium, ocadaic acid only works on PP1 and not on PP2A, so that would be something still to try. And in citrahymina, it is known that ocadaic acid speeds up the initiation of division, and also the division itself happens faster. And there's a different drug used to inhibit PP2A and tetrahymina that leads to the entire disruption of the filamentous actin structures, but it doesn't affect microtubules. So I will be very curious to see what it does in Stentor, because disrupting, if you imagine that filamentous actin kind of helps Stentor to kind of maintain its shape, if you disrupt it, you can expect to see those folds. So this is like future directions that I'm very much excited about. And so in addition to PP2A, of course, I plan to look at other protein phosphatases in Stentor and look at what sort of functions they have in the cell as well. And what is known about the different protein phosphatases in tetrahymina and paramecium is that they control, there has been reports of the fact that they control dyneins inside the cilia, and also cytoplasmic dyneins, maybe. So that fits well with the fact that my second phenotype is actually created by RNAi of cytoplasmic dynein. And as you know, cytoplasmic dynein is a wonderful, very huge molecule composed of multiple domains. And in paramecium, it has been shown, there's a very beautiful work that has proven the fact that cytoplasmic dynein in paramecium is actually responsible for transporting vesicles along microtubules in the cell. And also it covers the vesicles that are not attached to microtubules so that they can be transported. So that looks really exciting to me, because I'm really curious about how transport of vesicles is actually organized in Stentor. And so specifically what I'm going to be talking about next is about the RNAi of cytoplasmic dynein intermediate chains. And so when I perform the RNAi, what I see is that, first of all, the cells get much smaller over time. So this is a normal Stentor, and this Stentor has knockdown of cytoplasmic dynein intermediate chain. And second, what I observe is this interesting accumulation of vesicles that are actually, they might contain stentorin, which is the molecule that creates the blue color that Stentor has. And it looks like the color is absent from the tail, so I'm very curious about the nature of those vesicles. And there's indirect evidence that dynein in Stentor might be walking towards the tail, and the fact that vesicles are accumulated in this region and absent from the tail would make sense in that context. And in addition to performing RNAi, I just use Cilia brevin D to get to the same kind of point of inhibiting cytoplasmic dynein. And what I have observed is, first, the loss of cell polarity. So this is a control, and you can see that here the cells are much shorter and basically deformed in different ways. What's kind of really spectacular is that after about an hour of treatment with Cilia brevin D, what you see is basically cells explode. Like all of them will explode eventually, and this is how it looks. Within 2 minutes, a cell goes from being a cell to being a cloud of something, in the most dramatic way. And I'm very curious about this process because, as you can see, the cortex itself gets entirely destabilized. So I first thought maybe there is some sort of tension that is created inside the cell and it just creates an explosion, but you can see that actually this tail region of the cell, even when it's just like a flat surface that cannot be pushed from within by any like additional tension, you can see that it still dissociates itself until there's nothing left. So how would dynein be responsible for that is definitely something I'm very curious about. And when I'm thinking about like what can my cytoplasmic dynein intermediate chain be like, obviously RNA was for intermediate chain and the inhibitor was for, in general, for cytoplasmic dyneins, but basically thinking about all of this and trying to make sense out of what I see, there's this beautiful work in tetrahymena that talks about cytoplasmic dyneins in tetrahymena. And so this time, this work is about the heavy chain of dynein. And what you can see is there are two different genes for dynein heavy chain in tetrahymena, and one of them is responsible for phagocytosis. So in this case, you can see that the cells haven't phagocytosed fluorescent beads, and the second one seems to have a more drastic effect, like very different basically effect, because first of all, the cell shape is very, very perturbed. And secondly, which makes me really also very curious about this, is that actually both of those proteins affect cell size. So in the case of dynein heavy chain first, you see that the cell sizes become smaller, and this is exactly what I see with RNAi of my intermediate chain. And on the contrary, when in the experiments, when there's knockout of the second dynein, cells actually are becoming bigger and there's more variability in cell size in general. So this is something I would love to look at in Stentor as well, and it looks like my phenotype might be related to the phenotype of the first protein, so I will definitely be looking at what is happening with phagocytosis in Stentor. And so in summary, the two main phenotypes I was talking about today is protein phosphatase 2A, and as you have seen, it's important for maintaining the proper cortical row organization and in general for cell shape, cell morphology. And secondly, a cytoplasmic dynein intermediate chain is very important for Stentor growth and for the possibly for cytoplasmic transport inside Stentor. And I would like to thank my wonderful lab, our amazing advisor Wallace Marshall, who is here wearing a t-shirt that has Stentor color, of course, and the wonderful Stentor team we have in the lab, and Mark Slobodnik, who used to be in our lab. And you will hear from Sarah and Athena today. And thank you very much for your attention.
Hi. What's the extent to which that the microtubule rows turn over? Because one thing I wasn't clear from your RNAi is, are you forcing the cells to actually go through a cortical rearrangement, either via cell division or cutting? Because if those rows are really stable, I guess you might expect the phenotypes not to be that severe if they're not like forced to turn over. So can you comment on that?
Yeah. So in general, the microtubule rows, thank you for the question, they are very, very stable. They are acetylated very heavily, and so they're quite stable. To address your point, I was actually cutting the stentors in both of my phenotypes in two and looking at the regeneration of those stentors. And basically, what it looks like so far is that they do regenerate at the early stages of the phenotype development. They do regenerate to the same extent of looking weird as the original cell before it was cut. But at the later stages, since both of my phenotypes are actually lethal, at the later stages, basically whenever I cut them, they die very quickly because the original cell would have died as well. And in addition, they actually do divide also during the entire RNA process. It takes multiple days. So there is cortical row kind of rearrangement and kind of addition of microtubules going on. So this process is working, but somehow the actual organization of it is not working.
I see. But can you predict from either the cut part of the cell or the part that would rearrange during cell division, or is that part of the cell more screwed up, for example, than the parts that were not regenerated? Just so that, because that's where the demand for new organization or new transport, whatever, would be happening.
Mm-hmm. So far, I haven't observed the difference. I'm still trying to kind of really pinpoint like what would be the effect, but it looks like both halves regenerate kind of to the same extent of being weird. So,
Got it. Thanks.
But it's a very good question. Thank you.